Yeah, data does, especially last week.
Did you bring brownies?
Yes, I did. Yeah,
More brownies.
yes.
Well, good evening, everybody. I'm Dan Sivek, and on behalf of the Friends of Schmeekler Reserve, I'd like to welcome you to the first talk in our Research Sampler Series. And I believe I'm correct, someone can correct me if I'm wrong, that this came about when in one of our board meetings somebody said, what are those crate-like things in the water on the west end of Jonas Lake? And I didn't know the answer. I'm not sure what it was even. What are those things around?
Those are for the weevil study?
For the
Yeah.
weevils that are eating
You're raising milfoil.
the milfoil in the lake. So it's part of a long-term experiment.
Right. Well, the discussion kept going then on research. Well, what's this? What's that? What are those traps for out in the woods that you see? What's that? What's inside that fenced area over on the other side of Michigan Avenue there? And we thought, well, if we don't know these things and we're seeing them, we thought that maybe you folks would also be interested in that. So that's how this idea got started of a research sampler series. These talks are going to highlight some of the research projects that faculty, staff, and students here at UWSP are doing in the reserve. And in addition to tonight's talk, if you want to take a mental note, we have two additional talks coming up. On Tuesday, February 10th, the topic is flying squirrels. Research being done here on those, and apparently there are both the northern and southern races or subspecies of flying squirrels found here in the reserve. The following Tuesday, February 17th, the topic will be white-tailed deer, and if you live anywhere near the reserve or walk through the reserve, you are very interested in that topic. I think I have some of the deer here come to my place a mile away and munch my shrubs. And to give you a brief idea of how much teaching and research goes on here in the reserve, in the 2013-14 school year, so not this school year, but the previous one, over 5,000 students spent over 11,000 hours either in classes or doing research here in the reserve. And of course, some of those were probably counted twice because they were in different classes that were here. Let's see, in addition to the Research Sampler Series, the Friends of Schmeekler Reserve sponsors the twice yearly candlelight hikes, the last one this fall. How many of you were on that? Okay. Not surprising, the count was over 2,200, I believe, for last fall, so it was very big, very popular. The next one is coming up Friday, April 24th.
Friday, May 1st.
Oh, okay. I saw a different date. Friday, May 1st. Okay, so please come to that. And this year, one other project that the Friends Group is undertaking is there's going to be a renovation of the Trail of Reflections, or Reflections Trail, here in the reserve, and we hope that maybe you can get involved and help us out with that. If you're already a member of the Friends Group, thank you very much for joining and contributing, and we look forward to your future involvement. If you're not, we invite you to join. There are brochures, I'm sure, back there on the table, and you can talk to any of the board members. Raise your hand, board members. Okay, if you're not a member, you can talk to any of us or the Schmeekler Reserve staff. Ron Zimmerman's the director, Jim Buchholz, the assistant director, and Megan Espy is the outreach coordinator. So talk to any of us. We'd be happy to talk to you about the Friends Group.
And Carly.
And Carly.
Oh, I'm sorry, and Carly. Carly is the graduate assistant here at Schmeekler Reserve, and who's also doing research. What's your topic, by the way?
Mine's on assessing the feasibility for expanding our educational efforts, so this falls in, kind of in line with that.
Very good. Sorry about that.
Education, no, that's okay.
All right. Let's see though. I think that's about all I have to say about the Friends Group, unless any of the other board members or staff would like to say anything. Hearing nothing, then we'll start with the reason that you all came here tonight. So without further ado, our topic tonight is Wildflower Census of Schmeekler Reserve. And our speakers, we have two speakers tonight. Dr. Brian Barringer, who is assistant professor in the biology department here at UWSP. He earned a PhD in plant ecology and evolutionary genetics at Cornell University. And at UWSP, he teaches an introduction to plant biology, general ecology, and plant ecology, among other courses. And he's originally from Northern California. Tanya Copas, our other speaker, is originally from Merrill, Wisconsin. She's currently an academic department associate in the biology department here on campus. And previously, she worked as an adjunct instructor in the bio department teaching general biology labs. So please join me in welcoming our first speakers in the research sample series. Looking forward to it.
Thanks so much, Dan. Thanks, folks, for coming. Appreciate you coming out. I was going to say the really cold weather, but actually it's warmed up, so surprisingly so. I'm not from Wisconsin, so I assume that means summer's here. It's above freezing, or at least it was above freezing. I was laughing as I left the house. I thought only in Wisconsin would I wear a sports jacket and hiking boots. Anyway, I'm Brian, and I'm just going to sort of introduce what we're going to talk about here briefly, and then I'm going to give the reins to Tanya. We're actually going to talk about two very different, frankly, studies. The first of which is sort of up here on the overhead, this Wildflower Census that we began last summer and that we will continue on at least this summer and probably beyond. And Tanya has really sort of spearheaded that project. And we talked about this with the Science in the Northwoods. Was that the name of it? Yeah. In Boulder Junction, a few months ago. Now, something like that. So anyway, so she's going to, I don't want to steal her thunder, but she's going to talk a little bit about basically what we did last summer and where we think we're going with this. I want to emphasize that through this, we hope to keep things as informal as possible and absolutely invite questions and ideas and opinions. And in fact, I'll highlight a couple of points this evening where we're actively looking for input. Again, this is ongoing research, and so we would love to hear your ideas if you can think of ways that we can improve what we're doing. And so anyway, so she's going to talk a little bit about the Wildflower Census. Once we're done with that, I'm going to talk probably just for 10 minutes or less about what I hope will become a long-term study here at Schmeekley looking at the phenology of mostly tree species around the reserve. And so we spent a great deal of time last fall tagging 300-ish trees, again mostly trees around the reserve. And I'll talk a little bit about why we did that and where we would like to go next with that work. And I sort of got ahead of myself. I did want to make a point of not just thanking you all for coming, but thanking those of you that are affiliated with Schmeekley in particular for helping us along the way. And so, this Tanya and I are up here standing in front of you, but there have been plenty of folks that have contributed to this, as well as a few students. And I think she'll mention the students that were involved in the wildflower work, and then I had a student working with me last fall, and the goal, of course, is to maintain that involvement with students in this work. So anyway, so I think with that, I'll just sort of hand it over to Tanya. Again, we're going to probably go a bit short. I mean, actually, we got a bit of a late start, so it'll probably work out just fine. This is a work in progress, so we don't have lots and lots of data and lots of results to share. But on the other hand, I think it's exciting and that we can give you some insight into what we're up to now and again, maybe get some feedback and some ideas from you all on ways that we can improve this work. So I would encourage you to sort of interrupt us, ask questions as we go along. Tanya?
Thank you. Maybe turn some of these, oh, you're happy. All right, well, thank you for having us here tonight, and I guess I would like to take a minute and thank you to the Friends of Schmeekley Reserve, not only for being interested in supporting our research, but for supporting and advocating for the reserve as a whole. I think we're all here because we have a love for this wonderful asset to our community, so thank you. Thank you also to Megan Espy for organizing this event. I think it's exciting to share what we're doing in the reserve with the community. And I thought you were going to make a joke, so I had something to say along the lines of Brian was going to...
I can do it. I was going to make a joke that, you know, this is work in progress, so we're setting the bar pretty low. And so the good news for you guys is that for the rest of the talks this season, I suppose, it's sort of all uphill from here in terms of quality, right? So we're starting low. I mean, come on, flying squirrels, I mean, that sounds a bit sexier than plants.
So thank you for lowering your expectations.
Yeah, that's right. Yeah, the expectations.
All right, well tonight, as you know, I'm talking to you about the Wildflower Census that we conducted last summer in Schneekly Reserve. I worked on it along with Brian Barringer here, as well as two undergraduate students, Michaela Downey and Emily Shafkin, who I guess have now both graduated and moved on from UWSP. Before I talk about what we did in our study, I think it's important to give the study a little bit of context. Where did it occur, and why does it matter? Why is it important? Hopefully you all know that we are located in central Wisconsin right now. The star on the map indicates Schneekly Reserve. And the reserve is located in an area that's known as the tension zone. The tension zone is defined by the types of forests and where these two types of forests meet in the state. So in the northern part of the Wisconsin, these forests are generally called the northern mixed forests. And in the southern part, we have our southern broadleaf forests. These two broad forest categories adapted to different soil types and climates over time. They meet in this area in central Wisconsin called the tension zone. So we see plant and animal species of both northern and the southern regions here. And in fact, the tension zone is known to be the approximate northern boundary for many plant and animal species. Naturally, because we see species from both forest types here, there's a higher diversity of plants, animals, and other organisms found in this area. So studying and doing surveys like this in these locations is particularly interesting. Scientists are predicting that this tension zone is going to shift north as the globe continues to warm due to climate change. So we might anticipate to see some of those species boundaries or borders move north as well. Again, I wasn't quite sure of who my audience was going to be tonight, so I'm not sure where to pitch this. I'm guessing most of you are quite familiar with this map of Schneekly Reserve. Of course, we're up here in the visitor center right now. And you know that it's a 280-acre woodland, grassland, and marsh complex located at the north end of the UWSP campus. The reserve, the land here historically or during pre-settlement times is known to be a wetland area. I think Joshua Hathaway conducted some surveys in 1836, and most of the area was wetland. Over the next century and a half, a variety of land usages have been seen on these properties. There was a lot of logging that took place in the mid-1800s and farming and grazing in the late 1800s and through the mid-20th century. Most farms were abandoned by the early 1950s due to infertile soils. And by that time, I think UWSP was interested in the land and studying there, and eventually Schneekly Reserve was established in 1975 as an environmental laboratory. A couple of notable features here. Lake Jonas, as you probably know, was a man-made or excavated lake, I believe in 1974. And there's also the recently restored Moses Creek area here as well. Because of its designation as an environmental lab and because of its proximity to our campus, Schneekly Reserve really has an excellent potential to serve as a platform for long-term studies. And in order to support those studies, we need a lot of baseline data. We need to know what plants and animals are there, how many are there, where are they located within that reserve. And some of those studies have been done. I believe there are species lists for trees and shrubs, birds and fish for the reserve. But when Dr. Barringer came to UWSP in 2013, he quickly discovered that there was no wildflower species list for the reserve. So naturally we thought that would be an exciting thing to do. I'd do a great service to Schneekly Reserve and the people who come here and enjoy it. But I think we quickly realized realized that the study would also serve as important baseline data for potential long-term studies, which will be increasingly important in the next century as scientists predict lots of biotic change due to climate change, invasive species, and habitat loss. So what did we do to come up with this list? During the summer and fall of 2014, we visited 126 plots that were established by a previous study. We visited each one of these plots 3 times, in early summer, late summer, and then once again in the fall. So on the map here you can see, kind of the rough outline of Schmeekley Reserve here, and each one of these marks one plot within that reserve. We located each plot by using GPS coordinates and a metal detector. The plots are marked by metal stakes that have been driven into the ground. So some of them were quite challenging to find, especially in the wetland areas, and on a few occasions we had to just do our best and go with the GPS coordinates. Once we located each plot, we established a 10-meter radius around that plot, and you can see Dr. Barringer as well as Michaela in the photo here doing so. And once we did that, we identified and inventoried all wildflowers within those plots. When I say wildflowers, we cataloged herbaceous flowering plants, so non-woody species, no ferns, and what am I leaving out here? Thank you. And we did not do grasses, sedges, or rushes, mostly due to time and logistical constraints. So in addition to identifying those, we also estimated the populations for each species within each plot. We ranked them from 1 to 5 by the number of individuals designated on the slide here. And we also recorded phenology data. What was the plant doing at the time that we identified it? Had it not flowered yet? Was it in flower? Was it setting fruit? Or was it senescing or dying off? In addition to cataloging each wildflower species within each plot, we also inventoried any additional species that we found on the trail sides and in between or outside of those plot sampling areas.
Elsewhere, etc. But another consequence is that the phenological schedules of the species that are currently here might change. And so by phenology, what I mean by that is the schedule. I mean, you can define it in a variety of ways, but I typically define it as the schedule of life history events, or important events, I guess you could say, that an organism exhibits throughout its life cycle. So for example, if we're talking about a red pine, when do the buds first appear on that pine? When do the cones first appear, et cetera, right? So, it's a bit of a loose, or the categories that one might quantify are a bit loose and so on. But I was interested in this basic question. I mean, can we start a project now that may indeed become a long-term project, 10, 20, 30 years or more, gathering data now on the basic phenological schedules of plants here in Schmeekley, continue this annually, year after year, and then have something interesting to say, maybe 10 years from now, maybe 20 years from now. And so that's what I wanted to talk about just briefly here. The backdrop, I should mention, behind all of this too, is as a new faculty member here, I'm always on the lookout for ways that I can incorporate students into my research. And so one of the things that I thought right off the bat is, well, look, if I'm going to monitor the phenological schedules of a bunch of plants over at Schmeekley, maybe I can actually have students do that as part of a course where each of them, for example, I teach general ecology every spring. There are 120 or so students in that class. If I had a few hundred trees, I could sort of assign a couple of trees to each student, make them responsible for that tree or those trees, and then they could visit those trees throughout the semester and monitor what the tree is doing, essentially. I learned pretty early on, in fact I think Kendra was the one that told me about this, Project Budburst, which is this fantastic, and it turns out has a local connection, I'll explain in a second, this fantastic national citizen science project going on, where they are interested in collecting data for phenology, for any plant you care to send them data on, from anywhere within the United States. And so I contacted them because I thought, well, this is an obvious way that we could use this data, not only to say something interesting about what's going on in Smeakley, but maybe at the same time to contribute to a larger data set, feel like we're sort of contributing to something beyond just Smeakley. And also, again, thinking about my students and how they might feel like they're part of something bigger if they're uploading their data to this national effort, if you will. So I reached out to them and I contacted them and said, look, so if you read through their information, a lot of what they do is just your average person who has an oak tree in their backyard and is interested in contributing data to their data set will set up a quick account, it's easy to do, and just upload their data, just as sort of something fun to do. So I contacted him and said, look, I've got, potentially, 120 students every spring and a few hundred trees, and we could, do you have some easy way for us to sort of upload all of this data in mass? Are you willing to work with us, etc.? The short story is they were thrilled to do so, and in fact, ironically enough, the person that I ended up talking with, Sarah Newman is her name now. I guess it used to be Sarah Braun. I don't know. Does that name sound familiar, Ron, or anyone else? So she graduated from UWSP, I don't know how long ago, but I think it was probably 10 years. And as soon as I told her about this, she goes, oh, on my very first weekend at UWSP, I, along with a bunch of other students as volunteers, went out to Smeakley and pulled buckthorn. And I said, well, you missed some, because it's still there. Yeah, so she was thrilled, and in fact, we need to sort of work out the details with her, but we're hoping to sort of use UWSP and this system as a sort of model, or they're hoping, I should say, to use this as a model for what they do for other universities to then upload lots of data en masse. And so I think this project here, looking at the phenology of again, mostly trees at Smeakley, has the potential to grow into something a little bit larger, hopefully with their help. But anyway, so that's the backdrop. So what did we actually do? Well, we just got started this fall. I had a student from my general ecology class last spring approach me about doing research, and I said, well, boy, have I got a job for you. Here's a hammer and a bunch of nails and a bunch of tags. And Ron and Jim and Megan were gracious enough to let us do this. We went around Smeakley, I should say he mostly went around Smeakley, and tagged 300, well, as I'll explain in a moment, 299 actually, trees around Smeakley. And for now, all we've done are a few things. I mean, nothing really exciting phenologically is going on in late October when he was out there doing this. And in fact, he wasn't even able to identify, he wasn't able to even identify a lot of the trees to species, but that will come in time. But for now, he started this data set where, again, we've got 299 trees. Again, I'll explain in a moment, it actually goes to 300, but we've got them all ID'd. For many of them, we at least have his best sort of guesstimate for what the species was. I mean, some of them I think are pretty easy to ID, but others less so. We've got the region within Smeakley in which they occur. Importantly, of course, we have GPS coordinates for the actual trees, although I can tell you from experience, and those of you that have used GPS, I mean, they only get you so far. They get you, to within, whatever, 20, 30 feet of the tree, and then you've really got to sort of hunt around and try to find the tag. So we've tagged all these trees. We did go ahead, and I thought, well, if you're going to mark these trees, we ought to gather some sort of data on something. And so we measured DBH. So diameter at breast height, during fall of this past year. Again, the idea is that we will do this every year, but importantly, what we will start doing this spring is revisiting these trees and start monitoring phenology. So things like when when do buds burst, right? So when do they leaf out? When do they produce flowers? Later on in the fall, when are they producing fruits? When are the leaves falling, etc.? All those important sort of phenological events. So that's the eventual goal here. I've hinted at this a couple of times. There are 299 trees. That's because tree, if you will, number 300, he decided as sort of a joke, and I think it's pretty funny actually. He tagged a telephone pole right out here on the street near the visitor's center. I'll show you a map here in a second, by the way. And we dubbed it, it's a new species, it's Telephone polensis. We're not actually sure the actual species, but the genus is Telephone polensis. We've got the DBH there, it probably won't change much over time. And the GPS coordinates.
It's a control factor for you.
It is a control, that's right. So presumably this won't change much, at least in the near future. Funny enough, this actually sparked a bit, so I had sent this out to the biology department. I said, hey, look at what my student did. And we gathered all this data, and oh, haha, he marked a telephone pole. Isn't that funny? Well, many of you, or some of you probably know John Hardy in our department, who manages our Biology 130 labs and our greenhouse and does any number of other important things in our department. And he manages, so in Biology 130, which is our Introduction to Plant Biology course, among other things, students have to learn, I think the list is maybe 150-ish or something species, something like that. Does that sound right? Okay. They have to learn a lot of local, important, sort of common Wisconsin plants. In fact, that's what we call the list, is the common Wisconsin plants. And so we have this website, by the way, to help them do that with all these photos. So John thought it would be funny to add this to our list. Just for the record, students actually are not required to learn this. But it actually has its own entry, and I encourage you to read this. I'm not going to read the whole thing to you, but it's really, really funny. John did a great job. It's a variable group of mostly medium-sized, non-photosynthetic, parasitic trees with straight truncate stems, etc., often with a pungent creosote. I love, for those of you that know something about botany, where does he say this? There's something about stolons here, which I thought was funny.
Slender aerials.
Yeah, there we go. Yeah, so no known sexual reproduction, but forms extensive linear colonies linked by slender aerial stolons, much like strawberries, right? So anyway, I encourage you to check that out. It's actually pretty funny. In fact, he went on to actually describe the anatomy of it. So anyway, it's kind of a nice addition to our study. Here's the map, though. So back to serious stuff. Here's Schmeekley. It looks like a mess, and I guess it kind of is. I mean, the student working with me, Brian Zimmerman, by the way, is his name, worked very hard. He started out over here in the corner, out by, I guess this is Division, right?
Yes.
So he started out here over in the corner and was just kind of haphazardly walking along. And then at some point, I think more or less in here, he realized that maybe he ought to have some order to things, and he started to kind of walk in lines back and forth. And so more or less, a lot of these data points, are kind of sort of in lines. You maybe have to use your imagination a little bit. But and then he waited until the very late fall when he could walk out to the island and actually marked a few individuals out there on the island as well. Not a lot of trees in the Moses Creek area. Again, I think the Telephone polensis, is right there, right out there on the street. Anyway, so that's where we stand with this study. Again, this is very much a work in progress. I hinted at the beginning of our meeting that I was going to solicit you all for ideas. That goes not only for the wildflower census, as we talked about, but if there are things that occur to you, either now or later for that matter, shoot me an email or speak up now, things that occur to you that we could sort of do with this. I mean, this is just getting going, right? So we will likely add more individuals to this study. And so did Brian meet with you? I know there were a few, Ron, there were a few like individuals. I think that you wanted tagged. Did he meet with you about that?
Yeah, he did.
Okay. So again, we need to look at the whole list and try to figure out what the species are that he tagged and whether or not they are a sort of representative sample of the tree species diversity here at Schmeekley. And if not, maybe fill in any gaps in that regard. But other than that, again, we're sort of open to ideas. If something occurs to you that we could do with this data along the way, I don't know what I just did. What did I do? There we go, I did that. Then I'd be all ears. Bob?
I got to briefly run. Loved it, very much so. Another powerful global force, which I'm sure you guys have thought of, but I didn't hear it, so I thought I'd mention this. And a more, perhaps a prompter of quicker change globally is urbanization. And it's a fascinating dynamic right now in regard to a whole bunch of animals, and of course, as they relate to plant distribution and whatnot. And I'm very fascinated by a bunch of my colleagues who are involved with studying the effects of urbanization on these kinds of scapes that are the interface of that very powerful global force. And the only reason I mention, of course, that's another larger context in which that you might find some more significance to place whatever it is you find and track across time.
Sure.
Because sometimes it's hard to actually separate the effects of climate change from what urbanization.
Yeah, of course, right, yeah.
So, anyway,
So by larger global force, you mean climate change is what you're talking about.
Mm-hmm.
Yeah.
And I don't know if anyone noticed it, I go through here quite a bit on a daily basis. This is the first year that I actually observed on a daily basis fledgling ravens in Mercer, which I was really tickled by. And I don't know if you'd ever seen them before here, but that was really cool. I've seen some pretty neat stuff around here. Those fledgling ravens, that's awesome. And I've often thought that Ron...
Do they normally occur south of here or north
Well, no,
of here?
they occur statewide, but then there's another urbanization factor that as some of these species acclimate in some ways, and obviously this is a successful nest somewhere close by, given the timing at which I was seeing them and the regularity with which I encountered them. And I've never said this to Ron, but I say to all my students, I have a fair amount of plant background, so I'm going to say this quickly. This is a really cool place, this Schmeekley Reserve. The diversity here is a tribute, I think, an awful lot to whether it's something you specifically render as an objective or it's inadvertent. It's phenomenal, some of the diversity that I come across here. And I've searched throughout the entire state looking for all kinds of things, mostly birds admittedly. But it's fascinating to me, the diversity that's here. And not to, one last thing, I'll stop. The buckthorn.
Sorry?
Buckthorn automatically gets bad press, and I get that.
Yeah.
But from my standpoint, and of course I study a lot of avian ecology, I've been misnetting songbirds right on the edge there on the right-hand side. Man, the diversity of songbirds that come through, and every one of them are pooping out buckthorn berries. So just, it may be something you don't care for, but I assure you that a lot of native species are coming through, and I'm sure you're well aware of it, but I didn't realize to what extent that was happening until I started misnetting songbirds here. So anyway, I gotta run. Love the talk. Keep it up. Excellent stuff. Thanks for inviting me.
See you later. Other comments or questions?
If you want to add to the species list that you have for the flowering plants, you might corner Bob Freckman sometime and ask him about some, you can get a couple of highly endangered species that he doesn't tell people about.
Oh, okay, yeah. Well, yeah, I mean, for example, I know that sundews are here, and I don't think are they on our list? Or they are on our list. Okay, well, never mind, scratch that.
I knew where to look.
Okay. Yeah, because I know that they're in a secret area, but someone along the line, somebody had shown me those, yeah, we'll be sure to talk to him about that.
Yeah, he has just a few that, unless you ask him specifically, he probably won't.
Well, again, the nice thing about bringing Emmett on board is, he knows the plant. He's a walking encyclopedia as well, and so I'm quite sure that between the two of them, they'll be able to look at our list and say, well, you don't have this, and I know that occurs at Schmeekley, for example, and then we can go out and try to find it and get a voucher specimen. So, yeah, we learned pretty quickly. I mean, neither of us are taxonomists per se, and we learned pretty quickly talking with folks like Emmett and Bob that they weren't going to believe a word we said unless we had a plant to show them, right? So, I mean, you know, we can claim all day that species X occurs at Smeakley, but unless we have a voucher specimen, that means squat to them. So that's one of the reasons why we need to collect voucher specimens this summer, in addition to hopefully adding to our list. Yeah?
This is sort of extraneous, but I was coming through one day and there were some little boys whose father wasn't near them, and they were pulling up wildflowers it was by your thing, Jerry, by your gazebo thing, and they pulled up like 20 of them, and I stopped them, but they had a bunch of roots, and I was thinking, you know, that's probably a problem for your census, that there are people who come through and do that kind of thing. I mean, they were.
It could be. I mean, I would say, one thing I would say about that is that our plots, at least, our study plots, are off the trails.
I see.
So that I think would help. But of course, ultimately, we're interested in what species occur here, regardless of if they happen to be on our study plots or not, right? So I would say there's sort of the, I guess for lack of a better word, the scientific side of things where we've got these defined study plots and we can look for changes over time within those study plots. But ultimately, we want a list, you know, of the species that occur Smeakley. And so, and some species only occur trailside and or where there are light gaps and things like that, don't occur under the forest canopy. And so it's quite possible that, you know, a good example would be Campanulastrum americanum, the American bellflower that she mentioned, which I've actually worked on in the past. I was very surprised that it occurs here as well. And I can tell you just from my own observations, not here, but elsewhere, that, you know, it pretty much only occurs in highly disturbed environments. It doesn't occur in the middle of forests. It occurs on road cuts and places, trail sides and things like that, where little kids are going to yank it out of the ground. Now, with Campanulastrum, it's a weedy species, and I'm quite sure it will recover. But other plants maybe less so. And so, do you know what species it was?
No, I'm not any good.
Yeah. But yeah, I think that's one reason. And Tanya, I did this. I mean, wandered around, in addition to the plots, just sort of made a point of wandering around and recording species that we missed, you know, but ultimately it's impossible to know when to say enough is enough, right? There could always be a species out there that we just haven't seen. All we can do is keep at it and hopefully get to the point where we can look anyone in the eye and say, look, we've got 98% of everything that's there. You know, there may be a rare species that we don't know about or whatever, but I think that's the goal. And I think we're getting close. Yeah.
I just have a question. Is it legal for the public to wander around?
Is it legal for the public to wander around Smeakley? Is that?
Yeah, I mean to go off the trails.
Well, I mean, somebody else should.
Yeah, we encourage people to stay on the
Yeah.
trails, but if somebody has a specific reason for going off the trail, we don't discourage that. If somebody's biking off the trail or something, we try to correct them, our rangers do, and so forth. But for the most part, we want people to feel that they can wander off the trails and so forth, because it's a rare person that does that. Usually somebody has a real purpose if they do.
Yeah.
And in the Chilliwack lot, for example, we don't have any trails because it's used for class use and so forth, so we don't encourage a lot of people to move through there because it's a lot of student research.
I was going to say, you will see students. I mean, I bring my plant ecology, my ecological methods, my ecology and evolutionary biology students. We're all over here, you know, tromping around off trail doing things, you know, and so you'll certainly see people off trail, whether or not they sort of have formal permission or not. I think it just depends on the person. But.
Yeah, for example, if you're out here and you see something off the trail and you want to look at it closer, there isn't anybody that's going to correct you for going off the trail.
Yeah.
We feel that's fine.
Yes, this is kind of coming from the, sort of what I think Barbara and Nisha were getting at with the photo documenting and all of that, but I think this is a cool setting because, and it's unique because you've got this captive audience that's interested in wildflowers, and probably many of us are photographing or, you know, trying to ID or whatever. Is there something that you think would be useful with this new ambassadorship here of just us today.
Right, yeah.
But is there something that would be useful to you to have a community of people
Yeah.
contributing, you know, kind of similar to the whole citizen science movement?
Sure, yeah, thank you. I mean, I think it's a great question and a great idea. I think the short answer is absolutely. I mean, the trick is to figure out how to do it without our inboxes being filled with huge, you know, images, you know. So, you know, and again, I'm just thinking out loud here, but so someone may have a better idea. But one thought that comes to mind is, you know, we're getting to the point where by the end of this spring, hopefully, we'll have at least this preliminary document. We've got the document already. We'll have photos with that. We could sort of share it with folks. And, you know, so one thought that comes to mind is if you see the document, you think, that's a crappy picture. I've got a much better image of that species. I'm going to send it to them. Then by all means, I mean, that would be one obvious way to do it.
We could potentially compile a list of plants that we weren't able to get a photo of while they were in flower,
Yep.
or just
Yeah, 'cause we have
more
those
picture
as well.
quality.
Yeah.
And we could maybe make that available in some fashion.
Yeah, because, that's right, yeah, because we weren't able to get photos of any kind for some, and for some, we know that the photos we do have are terrible, so.
You could use us as a crowdsourcing.
Yeah, absolutely, yeah, so I think it's a great idea, so thank you. And I think that we'll make a point of doing that, and, you know, maybe, you know, Megan or someone else can sort of facilitate sending the document out to the Friends of Smeekly and anyone else who wants it.
We can put it on our website
So,
too.
yeah.
That would be great.
Yeah. I mean, again, we can share the document with you now. It doesn't have any images at all on it, but if you're just sort of curious what species are here, or at least the ones on our list, we're more than happy to share that. So.
Well, if anyone else has any questions, feel free. Otherwise, I'm sure you're going to stick around for a few minutes
you bet,
and talk to them. I think contrary to what you said at the start, I think you set the bar pretty high with your presentation here. We hope you'll come back February 10th for flying squirrels, February 17th for white-tailed deer, and springtime candlelight hike, May 1st.
May 1st, yep.
And also photographers, artists, and writers. We also have a contest that we would encourage you to get involved in. Barbara and Nisha, and who else is on?
Jackie.
And Jackie are on that committee. So if you're any of those, talk to these people. The next one is coming soon, right? The next round?
Next fall, it'll be so,
Yeah.
be preparing your artwork and photos and writing now.
All right. Thanks again, Brian and Tanya. Really appreciate it.
Help yourself to the refreshments, folks. There's lots back here.